Good afternoon. Thank you for coming to this talk. We have the pleasure to have Amy Osborne from University of Washington this afternoon to give a talk about her work on brain-computer interfaces. Amy took her PhD from Berkeley, had several years postdoctoralship in New York University, and now she is assistant professor and a lab director in the Department of Electrical and Computer Engineering in University of Washington. Without further ado, Amy, you have the floor.
All right, thank you very much. So my research is really interested in brain-machine interfaces, which the idea is very simple, that we can directly interface your central nervous system with the external world. So technology allows us to make measurements from your brain, and that allows us to get information out. But we also have the ability to do stimulation, where we can drive the activity of neurons in your brain with things like electrical stimulation or optical stimulation. And so this opens up a lot of exciting avenues for basic neuroscience studies, but also opens up ways to think about treating neurological disorders. So if you have someone who's paralyzed, for instance, because of a spinal cord injury, they're paralyzed because their brain can't talk to their muscles anymore, but their brain is still entirely intact and healthy. So if we could use sensors to record their thoughts as they're thinking about moving, we could send those signals to a computer, for instance, to allow them to control a cursor again or type. And so this, there's a lot of different flavors of brain-machine interfaces. You probably are very familiar with things like cochlear implants, retinal prostheses. My particular area of research is on the motor brain-machine interfaces, the idea of restoring movement to people who are paralyzed due to things like spinal cord injury and stroke. And so this idea has been around for quite a long time, and we've made some really exciting strides. There's clinical demonstrations in human patients, multiple sites now, one from Brown University and another from University of Pittsburgh, where they've been able to have great success with paralyzed patients who have a variety of different injuries like ALS or spinal cord injury, and they implant sensors into their motor cortex, and they can then record those signals and allow them to control a robotic arm to do really basic tasks of daily living, like reaching out and grabbing a cup to take a drink. But there's a really long way to go before these proof of concepts are going to be real viable clinical therapies. So just some examples of tangible challenges we face. One is that the performance is really far from what we can do with our natural limbs. None of these people are going to be playing the piano anytime soon with these brain-machine interfaces, doing complex tasks that require very high-dimensional dexterous movements, for instance. Also, the performance is relatively limited in terms of time. There's a lot of variability from day to day as subjects try to use these devices, and also typically performance sometimes declines due to issues in the sensor biology interface as well. And there's also actually a lot of variability in individual outcomes. So you'll, within certain parts of the community of BMI researchers, they have a term for this called BMI or BCI illiteracy. There's a subset of the population on the order of 20 to 30% that actually really struggle to ever use these interfaces successfully, and it's unclear why that might be. So these are just some examples of kind of concrete, specific challenges, but more broadly, the issue is that we have kind of very little principled or mechanistic understanding of how these interfaces work. This complicated system that we've created by interfacing the brain with engineered systems, we still kind of don't understand how that works at a more mechanistic level, and so that leads to kind of this problem of no design principles. So if you give me two patients, one with a spinal cord injury, one with a stroke, they likely need very different interfaces because they have different diseases underlying their paralysis, but I don't have any sort of good way to principally design interfaces customized to those particular people. And so one of the reasons that these design principles have been really elusive is because brain-machine interfaces are actually a lot more complicated than we originally thought. So typically people have always thought about this field kind of as a decoding challenge. We have neural activity in our brain that represents movements that we're trying to make. And so if we can build the right decoder, we could extract the perfect control signal to send to the cursor or the robot that we're trying to move. Problem solved, right? But this is actually just a subset of what's happening in a brain-machine interface. Once we have that control signal that we've decoded, it goes to a particular actuator that has certain mechanics and properties of how it moves, and then the subject has feedback about how it moves, and they can use that to update their strategies. So over the last several decades, brain-machine interface research has made kind of two critical observations. One is that the neural encoding, how neural activity relates to movements, changes between brain-machine interface control and arm movement control. So even if I have a decoder that could perfectly predict your arm movements, that doesn't mean that I can give you a brain-machine interface that's going to give you great performance. The second is that neural encoding actually can change with practice, and that subjects learn and change their strategies of how they use a brain-machine interface over time. And so these two observations initially were somewhat head-scratching from the field, but actually if you think about this system, kind of make a lot of sense. One is that we are not in a brain-machine interface where I'm, say, asking you to control a computer cursor. You're controlling something fundamentally different than your arm, right? A cursor is massless, it's frictionless. It has very fundamentally different properties than your limb. So it makes sense that the brain might change its encoding strategy and control strategy to move a cursor versus your arm. Also, the adaptation and learning that we see is obvious because this is a closed-loop system. Subjects have feedback, and brains are sort of optimized and designed to learn. That's what our brains do all the time. And so these observations kind of point to the fact that rather than thinking about BMI as purely a decoding problem, we need to think about it as designing a closed-loop control system where we have an adaptive element like the brain that can learn to control this new system. And so this is kind of the perspective that fuels all of my research, thinking about kind of how to optimize this new closed-loop control system that we've created in brain-machine interfaces. And I approach this from a couple different directions. One is kind of from an engineering perspective, given this new view, sort of rethinking some of the design choices that we make as engineers. So we can kind of treat the brain as some black box that we know is adapting and learning in some way. It has some encoding model we don't understand. And let's think about how we can design all the other elements of the BMI loop to deal with this black box. The other is motivated more from a neuroscience perspective, where we try to think about opening up that black box and actually studying some of the neural mechanisms of learning and control that are happening in the brain. And the goal, of course, is that these two research directions are going to inform each other. As we learn more about the brain, its encoding models, and control strategies, we can better tune our models as engineers to design the performance of the system and so on. Also, some of my work involves technology development because we're still kind of in a relatively new stage of neuroscience, and so we also are doing a lot of work to develop new tools to try to interface with the brain and make a lot of different measurements that will inform some of these studies. So today I'm mostly going to tell you about the first thread of research focused from an engineering perspective of kind of how this closed loop perspective informs our design choices of different elements in the motor brain-machine interface. So all of the work I'm going to tell you about uses a kind of common motor brain-machine interface paradigm in research. I work with animal models, so we work with monkeys. We do invasive recordings in their motor and premotor cortex. So these parts of the brain are very heavily involved in controlling your arm and hand movements, and also have really strong projections to your spinal cord. We put in sensors into the monkey's brain, and you can think of this as kind of just a bunch of wires. And we're recording from very small exposed tips on the ends of those wires, and that allows us to record the activity of individual neurons within the brain. And these neurons fire action potentials, and so most of the signals that we're dealing with are sort of these discrete events. They're sort of all or nothing. A neuron is either firing or not firing. And so these are discrete sort of Poisson processes are the signals. We use a decoding, typically really simple decoding algorithms, things like linear filters. Something like the Kalman filter, for example. And so what the Kalman filter is doing is taking the firing rate, so we take this Poisson process and convert it into a rate, and use that to predict the Kalman filter to map that rate of neuron firing into kinematics of a cursor on a screen. And we're typically using just a relatively small number of measurements, something like 20 to 30 neurons. The animals are then controlling the position of a cursor that's moving around in 2 dimensions on a computer screen. So position and velocity in this case are the kinematics we're decoding. And the subject has visual feedback of that cursor moving around. And so that closes the control loop. One other thing to note that's sort of, I think, often fascinating to think about is that these subjects are controlling this cursor without actually making overt arm movements. So these are able-bodied animals. They're not, we don't have disease models, but they're actually not moving at all. They're just thinking and changing their neural patterns of neural activity, and that drives the cursor around. Yep.
So you're using firing rates and going to kinematics. Are you doing that because that's the way the muscle activation works?
So we use firing rate basically because the current understanding of neuroscience is that likely neurons are kind of communicating via a rate model, that rate is kind of the language of neurons. And then kinematics is in some ways a convenience choice. I could give, a long lecture about how we still actually don't understand much about how motor cortex, the sort of representation scheme of motor cortex is still really unclear. Whether it's controlling muscles or kinematics is really hard to tease apart, in part because muscles and kinematic variables are heavily, heavily correlated. And so we don't actually know very much about the exact representation. What I can say is that we can predict kinematics quite well from patterns of neural activity.
When you say kinematics, do you mean position?
Yes, yeah, position and velocity, yeah.
Okay. So your model is you're going from firing rates to rate of movement and position.
Yeah.
Okay.
All right. So throughout this talk, basically, we're going to walk through kind of different elements of this loop and kind of visit certain choices that we make as engineers and think about how that influences brain-machine interface control. So the first question we're going to talk about is sort of the broad idea of how the loop itself, the properties of this control loop, are actually going to influence control in the system. So one obvious question when you're thinking, or property of a control loop, is the rate at which it operates. So in brain-machine interfaces, typically rates have been chosen kind of based on decoding perspectives, right? If you want to decode information, you probably will get better and better decoding outcomes if you sort of average neural activity over longer periods of time. And so, but from a control perspective, the rate at which someone is allowed to control a cursor is obviously going to influence their performance. And so what we did was design experiments so that we could actually test very specifically control and feedback rates within the loop and how they influence performance. And do this in a way that's separate from the rate of decoding. So there's a lot of different rates that happen in a brain-machine interface. If we think about the forward path, where we're going from neural activity to cursor movement. There's the rate at which we bin our spikes. Since we want to go from these discrete Poisson processes to a rate, we have to have some bin that we estimate that rate over. We also then, once we have our firing rates, decode the position at a certain rate. And then finally, we have the rate at which decoded positions are pushed to move the cursor. So this last step is actually really what defines the control rate. But typically people have kind of lumped all of these together, but even though these rates are actually associated with decoding. Because we're using a virtual system on the feedback path, the mapping from cursor movement back to subject feedback is just purely set by the screen update rate. So how often we're pushing the updated movement of the cursor to the screen to show it to the subject. So what we wanted to do was actually isolate this control rate from control and feedback rate from sort of this rate of decoding. And so we did that by using a rate-independent decoder that uses a point process filter. And I won't go into kind of the math and the details, but basically the idea is that rather than basically we have a decoder that operates sort of better fits the Poisson statistics of firing of neurons, and that allows us to decode neural activity sort of on a spike-by-spike basis. So it operates on zeros and ones. So we can now do our decoding on a very fast rate. In our case, we were using sort of 5 millisecond rates of decoding. And that means that now that we have a very fast decoding rate, we can subsample our control rates and do those manipulations independently of the decoder. Yeah.
Use all those neurons independently as inputs? So each of those 20, 30 neurons?
Yeah. So we have a model is fit for each neuron, and then they're all combined together to get the ultimate estimate of the position and velocity.
After the filter. So you do filtering per channel, and then you do the combination.
Yeah. And so with this point process filter where we can separate out the decoding rate from the control and feedback rates, now we can do manipulations of each. So in a first context, we have a fast feedback and fast control rate scenario where we're decoding on a spike-by-spike basis every 5 milliseconds, and every time we decode, we immediately push that to move the cursor, and we also immediately push that to the display to update the subject so that they can see that updated cursor position. On the other end of the spectrum, you could imagine doing a slow control and slow feedback decoder, where now the decoding is still the same. We're still decoding on a spike-by-spike basis, but we subsample the pushing that decoded position into moving the cursor. And same thing, we're only showing the subject the cursor when it's actually been moved. In the middle, we can separate feedback and control rates by doing a fast control and slow feedback condition, where now we're decoding and controlling the cursor at a very fast rate, but we're subsampling and only showing the subject on a smaller, slower time scale when we've actually updated the feedback about the cursor's position. Note that the inverse of this, where you'd be doing slow control but fast feedback, is sort of an unmeaningful test because you'd just be pushing static updates to the cursor, right? So this together kind of isolates all of the feedback and control rate conditions. For aficionados that are curious, in our case, fast for control means 200 hertz, so we're using 5 millisecond bins. Slow is 10 hertz, which is actually a really common rate that's used in a lot of brain-machine interfaces. On the feedback side, fast was actually just set by our monitor refresh rate. We didn't use ultra-fast monitors, so we just had 60 hertz. And then slow feedback is, again, 10 hertz. So now what I'm going to show you is the performance of an animal on a task in success rate, so the trials per minute that he's getting correct, for some of these different comparisons of these different types of decoders. And over here is just the relative change, so improvement in performance that you get by increasing either control or feedback rate. So first, if we compare the slow to fast slow condition, we're now isolating the effect of increasing the control rate. And so here what we see is that by increasing the control rate, we get an improvement in performance, so higher successes per minute. And this is actually very interesting because the subject doesn't actually have feedback about the fact that he's controlling the cursor faster, right? Yet he still gets improvements in performance. So this was initially slightly confusing, but if you look at the motor control literature of how we control our movements, there's actually a lot of evidence that we have feed-forward control mechanisms, that we have certain amount of modeling of how our movements happen that are completely independent of feedback. And so we can control our movements in a feed-forward way. And this suggests that the same type of thing is happening in brain-machine interfaces. And by allowing the subject to control faster, we're getting improved performance, even though he can't see the cursor moving that fast. So next, if we compare the fast-slow to fast-fast condition, now we're isolating the effect of increasing the feedback rate. And so here, again, we see an improvement in success rate. And this points to the fact that feedback also is really important in control, which is consistent with the fact that feedback control is very prevalent in the way that we move, and is also going to be true in brain-machine interfaces. And then finally, oh, yeah.
15 trials per minute
Yeah.
means in 4 seconds the monkey moves the cursor to the desired position.
Yeah. Yeah, so this is a center out task. So a trial is moving the cursor to the center, then moving out to a peripheral target, and we don't move him back to the center, so he has to also, it's sort of an out and back task. So he has to, in four trials in a minute, it would mean 8 out and back. Yeah, so they're going quite quickly. This performance is comparable to the rate at which animals do this task with their arms. And so then finally, if we compare the slow to fast, we see an even larger improvement in performance by combining the effect of control rate and feedback rate. And so this suggests that these two improvements in performance are actually somewhat independent of each other. So both feedback and control rate contribute to improved performance. And so finally, I wanted to mention that by combining this point process filter that allows us to do fast feedback and fast control, we were actually able to beat the state-of-the-art performance in brain-machine interfaces. So in blue, what I'm showing you is this same metric, success rate, for this point process filter with fast feedback and control, and in orange is the sort of previous state-of-the-art of a Kalman filter that's operating at 10 hertz. The feedback rate is still faster, but the control rate is 10 hertz. And so you can see that we get about a 30%, 20 to 30% improvement across 2 different subjects. And this is just an example showing you the trajectories of these center-out movements. And you can see that there's quite a remarkable improvement in terms of the straightness of the trajectories. And so obviously this improvement in performance is nice, but I think what I particularly like about this is that we were able to figure out where it came from, right? So we know that this 30% is a combination of improving the feedback rate, improving the control rate, and actually some statistical details of how the point process filter, by modeling the Poisson statistics of the neurons, we also actually get sort of an improvement in performance from there.
Can we say that it is personal because I see monkey J has 30% in
right. Yeah, so I do think that the individual variability across subjects is actually very interesting. We didn't, in monkey experiments, we're also often sort of underpowered in terms of the total number of subjects, but I think it's a really interesting question of whether, the degree to which subjects rely on feedback versus control, feed-forward control might actually vary across subjects, right? And so that might contribute to some of these differences. All right, so in this example, I hope I've shown you that, by not just focusing on decoding, but thinking also about the control in the system and sort of the rate and how that influences performance, we get significant benefits. But all of that work that I was showing you focused on where we already had a trained decoder. And I kind of glossed over how we trained that decoder at all, right? And so as I mentioned before, the encoding model typically changes between a subject controlling their arms versus controlling a brain-machine interface. And that leads to a bit of a problem of how do we actually train this decoder in the first place. So one possibility, if you have an unknown encoding scheme, would be to have an adaptive decoding scheme so that you could gradually over time learn the proper parameters of the decoder kind of in situ as the subject is using the interface. So this idea is I call closed-loop decoder adaptation. And simply, if you have subject using an interface in closed loop, you can look at the errors and mistakes that they make and use that to update the decoder parameters that you're using for control. And this idea has been used in quite a bit in brain-machine interfaces for a wide variety of different address a lot of different challenges. I've particularly focused on this in the context of kind of reliably and robustly learning this decoding algorithm when you have no a priori idea what the decoder parameter should be. So I want to sort of start from any random initial decoder parameters and get to stable final convergence, converge to a stable solution no matter where I start from. And this becomes actually an interesting problem in part because the subject's actually trying to learn at the same time as the decoder is trying to learn. So you now end up in a situation where the decoder is trying to learn what the subject's doing and the subject's trying to learn what the decoder is doing. And you have to make sure that these two learners interact synergistically. And so there's so the details of how you actually do this adaptive decoding become really important. One of the most important things we found ends up being the time scale. So one of the most important things is that the decoder has to learn faster than the subject, otherwise the subject starts chasing the decoder. And so, the decoder starts chasing the subject if you're not learning faster than the subject. And so one of the algorithms that we first came up with to do this, we call smooth batch. It's actually really simple, and from an kind of adaptive decoding perspective is suboptimal in a lot of ways, but it proves to be really powerful in this context of brain-machine interfaces. And so if you have a subject, a monkey, that's trying to use a brain-machine interface in closed loop, we can do a supervised technique where if he's trying to get this orange cursor to the blue target, and let's say that his actual velocity is going off in the wrong direction, since I know what the target is, where he's trying to go, I can infer his actual intention and use that as a training signal. And so now I can look at the activity in his brain and what I think he's trying to do and use that to create a new update of what my decoder parameter should be. And in this case, because we want to update the decoder quickly, we do this with just sort of a small batch of data, about 1 to 2 minutes. And then we push that to update the decoder. But because this is using a very limited amount of data, we don't want to just purely rely on this one batch. We want to kind of gradually evolve this over time with sort of a sliding window average. And so what we're doing is sort of slowly bootstrapping the performance over time. And this has a couple advantages. One, it allows the decoder to sort of update quickly, and it avoids things like subject frustration. But also, because we're doing this smoothing average, we're very robust to sort of noisy data. So let's say that a subject gets distracted for a portion of a batch, and you now have a really noisy, bad estimate. By doing this sliding average, we can kind of smooth a lot of those things out. So here what I'm showing you now is an example of what this adaptive scheme looks like in practice. So here is again this rate metric, events per minute. In black is successes, so when the subject's getting the trial right. And in blue is reach timeout, which is just an example of an error. And so when we start with a random decoder, he actually can't even make mistakes. The cursor is just sitting off in the corner of the screen, not doing anything at all. And so eventually, as we start adapting, he actually starts being able to make a couple of mistakes. He occasionally gets a trial right. And then he starts actually being able to succeed in the task, and performance starts to plateau. Importantly, if we stop adapting, the performance remains high. So this suggests that we've sort of converged to a stable solution. We've found some decoder that the subject can use readily. Really importantly, this algorithm is really robust and reliable. So we trained it across 56 sessions with a variety of different ways of creating this initial random guess, all of them being completely agnostic about movement and consistent with a paralyzed patient. And so in all cases, because we're not really using much information, our initial performance is terrible. But we get to the same final performance in all cases, so we're independent. Our final performance is independent of where we start. And we can also get there relatively quickly. So we hit maximum performance on average of about 20 minutes or so, even from any random initial seed. Yep.
So training happening in two stages. First, the subject has to learn where he needs to focus on, or what is the target. And once he's trained on that, then he starts moving the cursor?
Yes, so they are trained with the task. They know the task rules. separately from learning how to do the brain-machine interface control. So we train them on the rules of the task first with like arm movements, for instance.
Are some bananas involved?
Yes, research versions of bananas are definitely involved in the training. Yeah. And so once we sort of figured out some of the smooth batch was sort of a starting algorithm, and once we figured out kind of the general rules and how this worked, we then went back to the drawing board a little bit and refined this even further. So based on the observation that the rate of decoder adaptation mattered, we then went back and designed some new algorithms that work to update the decoder parameters really rapidly. So on every iteration of the decoder, you can actually use things like gradient descent and other strategies to update on a really rapid timescale. And this pushes our convergence down quite a bit. So we can now go from terrible initial performance to maximum performance in the order of just 6 minutes or so. And also, we also started thinking about the best ways to estimate a subject's intention. So you can also start thinking about making optimal feedback control models of how subjects are trying to control the cursor to actually get a more principled estimation of their intent. And this allowed us to improve the success rate of how quickly subjects are moving the cursor and without sacrificing any accuracy. So they're still making the same level of errors, but they're moving much faster. All right, so to quickly summarize this, you can use adaptive decoding to kind of deal with this issue of the fact that we don't necessarily know ahead of time what the brain's control strategy might be. And it's important, given that the subject might be trying to learn, the rate that the decoder adapts is going to be really important. And that we can do this really rapidly and we can robustly as well. But now we have an issue of how do we actually maintain this performance long term. All of those examples I showed you was for one example session where I trained that decoder on that day and then I tested it on that day. How do we start thinking about making a device that someone could come back to day after day and reliably use?
You said you're using a Kalman filter for the decoder?
Yeah, in that case it was a Kalman filter, yeah.
And have you experimented with other types of filters, like machine learning models, for example?
So because, not too much, and in part because of the fact that there's very little kind of correlation between how well a model predicts neural relationships between movement and neural activity. Doesn't really translate into improved performance. And so we haven't focused on alternative algorithms. One of the reasons I like the Kalman filter is because it's very interpretable from kind of a control perspective. It's very clear what signals are being sent and the mappings between neurons and control signals. But there's a wide space of algorithms that people have used. I don't think that the interesting thing is that really complicated networks, like even recurrent neural networks that people have used, have not shown, leaps and bounds improvements in closed loop performance, in part because of this issue that it's not actually a decoding problem. Yeah. Right. So if we're trying to think about maintaining performance, I will propose that one of the ways that we might want to do this is start leveraging the learning and plasticity within the brain. And thinking about that element of it, that this adaptive decoder is also dealing with an adaptive brain. So one of the reasons that we have a challenge with consistent performance across days is because we have measurement variability. So if we're putting these little wires into the brain to try to record activity of individual neurons, any movement between the wires and the brain is going to shift what signals you're actually picking up, right? Neurons are teeny tiny. So even micromotions will change your signals. And so we have this issue that we often have variability across days. So typically the strategy would be that we just retrain our decoder every day. If I'm not recording the same neurons, why should I assume that I should use yesterday's decoder? I should just start over again from scratch. And so this would avoid kind of declines in performance over time. And in fact, you can see that, if you do this, where this is the average performance on a task across days, where every day I've retrained the decoder, you can see that performance is generally quite high, but you also see that there's a lot of variability, right? you would be pretty frustrated if every day that you picked up your iPhone or whatever phone you have, your typing was worse or better depending on the day. And so this regular retraining sort of avoids declines in performance, but it doesn't actually help with variability at all. And so one possibility is that what we're doing is kind of disrupting the subject's ability to learn on longer timescales. And so, this would be in some ways equivalent to me asking you to learn how to play tennis, where every day I gave you a different racket that was slightly different weights or different sizes. And you can imagine that because we're constantly changing the decoder on the subject in subtle ways. And so one possibility is that we need kind of decoding strategies that are compatible with long-term learning phenomena. So a strategy that we came up with, we call sort of co-adaptation. And it's, again, a really simple idea that we have an initial decoder. We then do our decoder adaptation to try to boost up performance initially, and then we keep it fixed. And so we try to record as best we can stable signals, and we keep the decoder the same. And this will allow plasticity and learning on the side of the subject over time. But if we have a shift and measurement noise sort of leads to a reduction in performance, we can go back. So rather than starting back at the drawing board, we should just go back and make slight tweaks and adjustments to this. So if I've lost a neuron in my ensemble that was contributing to the decoder, I can swap in a new one, but keep all the rest of the neurons that we think are the same still in the ensemble. And so we can retain performance and kind of make just gradual shifts over time, and the hope is that the brain can actually track some of these slow, subtle changes over time. And so here I'm going to show you a video of what it looks like when we do this. And so this is day one with the performance with the initial random seed. So the subject is trying to get that white cursor to the green center target. You can see that he's really struggling to do that. Now this is the same day, but I've run my smooth batch decoder adaptation algorithm. And so now he's actually able to do the task, but he's still somewhat sloppy. You can see in particular that he sort of really struggles to actually kind of precisely get to the target and stop. That's one of his biggest problems. Now this is day 3, where we've left the decoder the same. The only difference here is that he's had time to practice and improve. So now you can see that performance is still sloppy, but much improved. And in particular, he's sort of more precisely getting to the target and stopping. And now this is day 13, where we've done gradual small tweaks to the decoder over time, and he's had time to practice. And you can see that performance is quite good, where we have very straight and quick trajectories between the center and the target. And this performance, again, is pretty close to what they can do with their own arms. All right, so this is just a graph quantifying the performance that you saw in that video. So percent correct over different days. So we start off with terrible performance, we boost it up using decoder adaptation, and then we can continue to improve from there over time. And all of these little blue dots are times when measurement variability sort of led to reduced performance, but we were able to do this sort of small tweaks to the decoder over time and maintain this consistent performance. And this also worked in the second monkey. One of the interesting things that we found when we did this is evidence to suggest that by incorporating brain learning, we could actually get better performance than we could with just purely machine learning alone. So here I'm showing you an example where on day 1, we sort of fully maximized performance as much as we possibly could on the first day. So he got up to 100% correct on day 1, and he was able to maintain performance over time by keeping the same fixed decoder and then occasionally tweaking it when we needed to. If you look, though, at the success rate, what you see is that over time he's getting better and better, making these movements faster and faster. He's also, if you look at error, he's actually reducing his error as well. And so this suggests that incorporating brain learning and long-term learning might not just would both reduce variability across days, but actually also give us better final performance if we allow the subject to learn and kind of master this skill of controlling the interface. If we now look at what's happening in the brain, it's interesting to think about, what is it that the subject's actually learning when they're controlling these interfaces? And sort of the short story is that they're kind of learning which neurons are contributing to the cursor movement and how to recruit those really precisely. And so what I mean, an example of what I mean by that is here I'm showing you an example of one neuron on a particular day. This is what we call a direction tuning curve. So this is the direction the subject's trying to move the cursor, and this is the firing rate of a neuron, that neuron, at the time he's trying to initialize a movement to go to that target. And we fit this with a cosine model, which is a very common model that we see in motor cortex neurons. And you can see that, the neuron sort of fires most for movements in one particular direction, and then opposite, for movements sort of 180 degrees opposite, it fires the least. If we look now at what this neuron does across days as the subject is mastering this task, the preferred direction, where this neuron fires most, is still the same target, but it's actually firing more and more, generally speaking. So the subject is learning that this neuron is important and making it fire more often. This is just another example of actually a neuron with an even more extreme case, where initially this neuron had no consistent relationship to movement at all. But over time with practice, the subject learned that this neuron is important for controlling the cursor and actually started making that neuron fire. We also see evidence that there's sort of faster and tighter temporal recruitment of neural activity as well, and these patterns become more and more stable over time. And this is really consistent with a lot of evidence from the neuroscience literature that this is kind of a hallmark of learning a skill. And so this points to the fact that, by neural and decoder adaptation can kind of work together in a synergistic way, and that brain learning might be really important for kind of creating robust interfaces and giving subjects kind of the opportunity to master the skill of controlling the interface. And importantly, that kind of what they seem to be learning in this skill is really which neural activity to recruit and when. Yeah.
The subject, each one happens here.
Both. So and maybe we can come back to this question because I've got, I think, a slide and a couple of slides that will maybe hopefully sort of point out, I think, what's happening in the big picture of what has to be learned in a brain-machine interface. You can kind of divide it into two things. And I think when you have decoder and in this scenario, the decoder is learning one thing and the subject is learning another thing, is kind of how I think about it. So let's come back to that. So one quick point I wanted to make is that some of our next steps for thinking about how to continue this research is this is all just controlling a two-dimensional cursor, right? And there's kind of obvious, interesting questions to think about how we would scale this to higher dimensions. So going from a cursor to something like our hands that has on the order 27 degrees of freedom. And this in part requires a lot of work that we're doing to try to think about how to really track and study high-dimensional movements. And in terms of motor learning neuroscience, we actually know very, very little about how people learn in high dimensions. So there's sort of interesting challenges both from the engineering perspective of how to design algorithms that are optimal and scale optimally, but then also kind of really open questions of how subjects are even learning in high dimensions. All right, so in the last little bit, I want to talk about, kind of go back to this question of, what is it that the subjects are learning, and kind of point to some of the new work that we're thinking about in terms of how to optimize this learning on the subject side. So in this sort of co-adaptive paradigm, one of the things I started realizing is that the subject actually in many ways is the bottleneck of this performance. And I'll hopefully try to explain what I mean by that in a second. So if we think about what's happening in brain-machine interface, where we're going from neural signals over to cursor movement, what we're doing is placing our sensors in some randomly selected part of the brain. And we have, thousands of neurons within your motor cortex, and we're arbitrarily picking some small subset of them to control the cursor. And then the firing of those neurons is going through some mapping of the decoder, right? In a linear filter, this is just a simple linear mapping that, compresses the dimensionality from the number of neurons you have into the number of control dimensions. So you can sort of break this big learning down into kind of two things. One is modulation learning, what I call modulation learning. And that means that the subject has to figure out how to make those particular neurons, the neurons I've highlighted in purple, fire reliably. If those neurons never fire, the cursor never moves, right? And if they never fire reliably, the cursor would never move. And given that we're kind of, arbitrarily placing our sensors in some random part of the brain, it's in some ways not obvious how the subjects are figuring that out. The second is a mapping problem, that given a particular pattern of neural activity that you're able to evoke, how does that relate to the cursor activity? And so I think of closed-loop decoder adaptation. The decoder learning is learning and simplifying the mapping problem, right? It's taking a given pattern of neural activity that a subject has produced and simplifying and learning the right mapping for a pattern of neural activity that a subject produces. But how is modulation learning happening at all, right? We don't actually know. So going back to the observations we made previously, the fact that the changes in neural activity that are happening are neurons firing more suggests that the subject is in some way solving this problem of figuring out which neurons are contributing to the cursor and making them fire more. So that's what they're solving. They're not dealing with the mapping problem because we're helping them with that via the decoder adaptation. They're figuring out which in the world of my billions of neurons in my brain do I actually need to make fire. And so going back to this kind of issue I talked about at the beginning of BCI illiteracy, right, that some subjects can never really learn to control these interfaces, maybe part of the reason why is that they are really struggling with this part of the problem. And this part of the problem is the bottleneck, right? Because we can have all of the smart machine learning we want, but if the subject can't produce the neural activity reliably, it doesn't matter. And so that has led me to sort of really think more about in detail about this problem and what it means. And so one of the ways to think about this and kind of how we might optimize it from an engineering perspective is kind of rethinking how we actually select our signals. So like I mentioned before, we're just putting our sensors somewhere in the brain. It's based on the fact that motor cortex controls movements, but we're not sort of more principally selecting these signals in any way. And given the fact that BMI is not a decoding problem, again, it sort of opens up the possibility of, how do we actually select these signals? And in addition to where you put your sensors, there's also the question of what measurement you actually make. So there's a lot of different ways that you can measure neural activity, right? We, in all of the data I've shown you so far, are putting electrodes into the brain and measuring activity of individual neurons. On those same electrodes, we actually also get other signals that people often call local field potentials. It's sort of low frequency activity that reflects summed activity of a lot of neurons in the listening sphere of this electrode. We can also do things like electrocorticography, which are less invasive, where we put electrodes on the surface of the brain. And we get similar signals where we're kind of summing up the activity of a larger group of neurons. And you can, continue to do this on a variety of different scales. You can not put it on the surface of the brain. You can put it on the surface of the dura. You can put it under the scalp. You could put it over the scalp. You can do this sort of field potential signal you can get from electrodes kind of placed any distance from the brain, right? So EEG is just a further zoomed out version of this signal. And generally speaking, in broad terms, the difference between something like EEG and electrocorticography is kind of the listening sphere, right? So you're getting, because you're further and further away, you're picking up neural signals from a larger and larger.
Spatial resolution is low.
Exactly, yeah. So you're losing, as you go from something like spiking activity to electrocorticography out to EEG, you're kind of spatially blurring your signals. In addition to loss of amplitude, right? Because the skull is basically an amazing low-pass filter that kind of cuts out all of your signal. Yeah. So if typically, so all of these different signals, spikes, local field potential, ECOG, EEG, all of those measures have been used for brain-machine interfaces successfully, but it's kind of still unclear which is the best choice. And they have different pros and cons. So thinking about kind of the two extremes, spiking activity is commonly chosen in large part because it's very closely correlated with behavior, right? We kind of understand in some sense, what spiking of neurons means, and it strongly relates to behavior. But we have this issue of kind of poor longevity and signal instability. It's also obviously highly invasive. On the other extreme, something like electrocorticography is typically thought, we sort of still don't understand a lot about how it relates to behavior. So kind of the encoding relationship between ECOG and movement is still an open question. But it's potentially longer lasting, more stable, slightly less invasive. And so, again, from a decoding perspective, the choice is obvious. If you talk to people that think about brain-machine interfaces from a decoding perspective, they say, obviously, spikes are the best, and we should go with that. Or, pick your signal based on what gives you the most decoding information about movement. But you can, again, flip this question around and think about it from a control perspective. And going back to this modulation question, which of these signals is actually easier for subjects to learn to modulate and control, and why? And I won't kind of go into the details, but there's a lot of reasons from kind of a controls angle that it's not obvious whether spiking or electrocorticography, as an example, would be easier for a subject to learn to control. And so I'm interested in starting to do some of these experiments where we kind of specifically look at how subjects learn to modulate neural signals and how that depends on the selection of your signals, both in terms of measurement modality, also where you put your sensors and things like that. Those are sort of new experiments that we're going to be starting in my lab, but I wanted to touch on the fact that doing these types of experiments actually required developing new technology. And so we had to develop a way that we could actually make some of these different types of measurements all within the same part of the brain, right? So if we want to do a direct head-to-head comparison between spiking and ECOG signals, we need to have the same part of the brain within the same subject, make all of those measurements together. And typical neural interface technology doesn't support that. So typically people put in one type of sensor, close everything up, and that's it. So what we did was develop an interface that allows for modular and flexible recording with a variety of different measurement signals, so that we could combine these different measurement modalities together. And so the general idea of our implant is basically, you can think of it as kind of an access port to the brain. I won't go into kind of the implant details, but it gives us chronic subdural access. So what we actually do is basically open up a window into the skull and also the dura, which is a covering over the brain, so that we can both see the brain and have access so that we can put electrodes in to make measurements. And we design the implant in such a way that it's really minimal. The chronically implanted portion of the interface is very agnostic to the measurement hardware that you're going to make. And so then that allows us to sort of stack things up. So you can think of our implant as like implanting the green Lego board base, and then you stack up Legos on top of the implant depending on what you want to make, what measurements you want to make. And so this enables us to do sort of very flexible recordings, both electrical and optical, since we have optical access to the tissue. And we can also I won't talk about it here, but we can also do causal manipulation. So we can do stimulation with both optogenetics and electrical activity. Or you can also think about doing silencing, where you can inactivate neural activity. So this is really exciting as an enabling technology to start kind of teasing apart the details of neural circuits and then how they relate to kind of this modulation learning problem in brain-machine interfaces.
What is the physical basis of optical recording? What do you record?
So there's several different optical techniques. One is, in this case, one of the things that we've done is intrinsic imaging. So you can think of it as fMRI, but on a much smaller, higher resolution scale. So you're measuring changes in reflectance of blood vessels that change as oxygenation. Yeah, exactly. So you can look at sort of changes in oxygenation in the blood. You can also, in animal models, obviously, you can inject GCAMP, which is an indicator where now neurons' expression of fluorescence changes with calcium. And so calcium is kind of one of the things that changes when a neuron fires an action potential, and so that allows you to pick up firing of neurons.
What is the diameter of this?
This is about 2 centimeters. Yeah. And so, so for those not super familiar with the brain, this is the central sulcus, sort of roughly in the middle of your head. It divides kind of the sensory parts of the brain in the back from the sort of motor and more cognitive parts in the front. So this is the motor cortex up here, and this is the sensory cortex. So in a monkey, this size scale covers multiple brain areas. In a human, this obviously, a monkey brain is about 10 times smaller than a human brain. So this size scale probably is more like one brain area in a human. All right, so with this capability of this sort of flexible implant system, it allowed us to design, as an example, an implant where we could do simultaneous electrocorticography, so recording from the surface of the brain, in addition to penetrating sensors as well. And so it's a little hard to see in this picture, but this is now that same brain that you saw before, but with an overlay of where we're making measurements. So these sort of bigger holes are where penetrating electrodes go through, and each of those tiny dots is an ECOG contact where we would be recording from. So we have 244 ECOG contacts and then 32 penetrating electrodes. And so this allows us to now make a variety of different measurements at different spatial scales within the brain. So we can look at sorted activity, where we're looking at individual neurons. We can also look at things like multi-unit activity, where this is kind of larger groups of neurons. These are, again, just action potentials, but it's picking up action potentials from multiple neurons that you're recording on the same electrode. We now have local field potentials, which is, again, this measurement from within the cortex, but sort of the summed low-frequency activity of neurons around the electrode. And then ECOG, which is that same type of signal, looking at activity of groups of neurons, but now recorded from the surface itself. And so there's obviously kind of very interesting basic neuroscience questions about how these different spatial scales relate to each other. But I'm particularly interested in, given these different spatial resolutions of measurements, which one is actually going to be the best signal for a brain-machine interface and why. Again, motivated from this perspective of the subjects have to learn to modulate the neural signals and create kind of reliable patterns of neural activity. And so it might be different depending on the way that you're measuring the neural activity.
The 100 microvolts scale is for both LFP and ECOG, right?
Yes, for both. So they're comparable.
So we lose 100 times of magnitude because of the skull. When you do EEG, it's a microvolt.
Yeah. All right. So to wrap up, I hope I've sort of convinced you that thinking about brain-machine interfaces not from a decoding perspective, but from the perspective of creating this closed-loop control system that the brain has to learn how to use is particularly useful for improving brain-machine interface performance. And so we can, again, kind of revisit a lot of the existing choices that people have made when we design these systems to try to improve the performance. And that includes things like adaptive decoding, incorporating co-adaptation, where the decoder is adapting along with the subject, other things like how you actually design your loop properties, and even how you select the signals And this is going to be really critical, I think, for making robust interfaces. Not just robust in terms of kind of long-term stability within a subject, but also across subjects. Because this allows us to sort of think about how subjects are learning these interfaces and optimize the learning process so that it's robust. And then, importantly, I think kind of insights into how subjects are controlling and learning brain-machine interfaces are going to be kind of really critical for creating these design principles that I alluded to before. So with that, I want to thank you for your attention, of course, and point out other folks that I've worked with. So all of the loop manipulation, decoder adaptation, and co-adaptation work is from my PhD with Jose Carmina at UC Berkeley. The multi-scale neural implants, the flexible implants that I talked about at the end, is work done with Bijan Passeron as part of my postdoc. And then we have a variety of different funding sources. So, thanks.
Thank you, Amy. That was an interesting talk. Colleagues, you have the floor for questions.
What's L'Oreal's interest in this?
Huh? Oh, L'Oreal. I got a Women in Science award from L'Oreal. Yeah. Yep.
One other question. Does the task actually get easier for the subject the more electrodes you have?
That's a great question. So the short answer, I guess, is that it's complicated. I think that my prediction is that there's a sweet spot of if you have too many electrodes at a certain point, especially given that machine learning is always somewhat suboptimal, you'll never get to the point where irrelevant signals contribute zero. And so there will be a point at which adding signals actually adds a penalty cost. But there is, in general, some benefit up to a point, I guess, of adding neural signals.
So is it true that the minimum number would be 2 for this task?
Yeah.
Okay.
Yeah, and it's...
But that's hard.
Not necessarily. So there's actually quite a bit of basic science research in BMI, particularly the things where people are looking at how subjects learn these interfaces, where they use really small populations of neurons, 1 or 2, or maybe like up to 4 or 5. And it's possible, and the subjects learn pretty quickly. Yeah.
If the subjects are human, can you ask them what did you think when you moved the cursor?
Yes. So in the clinical trials, there's definitely some interesting work where they try to use, the subject's ability to talk to try to learn a little bit more about what's happening. The interesting thing is that I think, sort of similar to how if I asked you to explain to me how you learned to play tennis, you would probably have you could tell me broadly what you were doing, but at the nuts and bolts details of how you perfected that serve, you'd really struggle to tell me. And I think there's very similar phenomena that happen in brain-machine interfaces where subjects will say vaguely that they started with a certain strategy. For instance, I thought about wiggling my tongue when I wanted to move up, and then I thought about wiggling my foot when I wanted to move left. But eventually that sort of fades very quickly, and they eventually can't tell you what they're thinking about.
So it never translates one-to-one to actual hand movement, for example?
I think every subject tends to have a slightly different strategy, and that actually relates back to, if you look at like decoder parameters and other things like that, you will also see that neural features that contribute to a decoder vary a lot across subjects as well. So people pick kind of different neural signals to modulate depending on their own personal preference. And part of what's really interesting in, thinking about the learning that subjects are doing is we like to think that we're optimal in learning, but we actually often get kind of stuck in local minima. And so it's going to be really one of the interesting things to think about is kind of how do we optimize that learning process to, help people find the best solution and not get stuck in based on whatever initial strategy they tried, that's going to bias where they end up in the space. Yeah.
So I guess related to that, how do you test that this whole feedback loop that it generalizes? So I'm wondering, in the example you had where you move your cursor, it looked like the target locations were discrete.
Yeah.
What would happen if you would rotate it
Rotate
by
it?
22 degrees?
Yeah, so we've done, I didn't include it here, but in, for instance, the point process decoder, we also did tests where we gave subjects arbitrary positions to move to and even gave them obstacles so that they had to not use a straight line path between the two targets, and the cursor control generalizes in that case. Generalization more broadly, I think, is a really interesting question. So one example is that we're talking about very controlled lab settings where the subjects are only doing this one task, right? Example of, trying to grab, this mouse while I'm talking to you, is another really interesting question of generalization that's an open one.
More
Yeah.
questions, go ahead.
Yeah, have you tried doing experiments using the visual cortex instead and just have them look at the targets?
So I personally have not, but people in the brain-machine interfaces community have. So there's a lot of questions about, could you do this control kind of from any part of the brain? And the interesting thing is that you can use something like visual cortex. In this case, it was done in rodents, so it was a slightly different paradigm. The neural activity was controlling an auditory cursor, since rats are not very visual animals. And so they were listening to tones and controlling the tone of the cursor with their neural activity. And so it had been demonstrated that you could do that in motor cortex, but you can also do that in visual cortex as well. And so in general, sort of the flexibility and plasticity of the brain is very, allows this to work with a wide variety of neural signals, which is actually really exciting because it points to the fact that, let's say I have someone who's paralyzed because of a stroke. They're paralyzed because their motor cortex and their motor neural circuits are damaged. And so it suggests that you could restore motor function, or whatever function has been lost due to neural damage by repurposing another part of the brain, right? Yeah.
Going back to the separation of control rate to feedback rate, I'm really curious at what kind of investigation there is done to the discrepancy in having a better result because of the control rate when the feedback rate does not change. Does it still make it more accurate because your control rate is higher? There are some fellow audio notes here, so I would compare it to the concept of upsampling for filtering to make it more accurate. Is it something like that maybe, or?
Yeah, so we haven't done, I think to really properly answer the question, we require some really carefully designed new experiments. But based on the data that we have, and we haven't done those, based on the data that we have, what we think is happening is sort of going back to this idea of kind of feed-forward control that's happening, where you're kind of launching the cursor based on the existing knowledge that you have, independent of other feedback that you're getting. What they're improving on and where the improvement in performance comes is actually related to holding at the target. So they are better able to kind of stop precisely at a particular point. And we think it's sort of, they're better able to kind of predict, or they're using the fast control rate allows them to kind of more precisely stop the cursor at a particular target. Yeah.
How individual are the decoders? What happens if you plug a decoder, a software from train it on one of the primates to another?
I haven't done that experiment, but I think that especially with these highly spatially localized signals, the generalization would probably be very poor, right? Because the heterogeneity of kind of how neurons relate to movement is really high. Whether that's true in, sort of more spatially average, less spatially precise signals, you might get more generalization because you're not trying to map one subject's brain, in fine resolution to another. But yeah, it's a great question. I think there has been some work trying to kind of apply things like ideas from deep learning, for instance, into brain-machine interfaces to deal with some of this, both robustness over time, but you could think about using it for subjects as well, where you kind of have a large library, and based on, past decoders you've seen, you could predict what the best decoder is for a new subject that you haven't observed before. But that hasn't been done.
It is because you have a very fine positions of the electrodes.
I think that's a big part of it.
Well, if you do EEG, for example, a set of electrodes over the same motor cortex, they will average in general.
In principle, yes. I think the question is whether all subjects use the same strategies, right? And so that's where the generalization is less clear, and you would probably need to do some amount of adaptation to customize the decoder to a particular subject.
Whatever signal you use.
Yeah, I think so, but it's an open question that I don't think, the literature doesn't speak to it yet, but that's my assumption.
What is your take on learning how to use the BMI? So you have a good adaptation system, you put the cap on subject which doesn't know, have never ever worked. Do we still will need learning, and how long it is expected to be?
So I do think that each subject is going to have to learn how to control the interface, because it is such a new thing for each particular subject. In terms of how long it will take, one of the things that I kind of glossed over is I showed these example learning curves that were on the order of 2 weeks, right, for one example decoder. But that one learning curve is actually part of a much larger learning curve. So in addition to the subject learning a specific decoder, they're actually also learning this bigger picture task of how to control the interface itself. And so it's sort of learning how to learn in some ways, you can think about it. And that time scale, I would say, is on the order of a month in these particular cases. Whether that would be the same for, a human subject, especially where you could, in principle, tell them more about the rules of how things work, I don't know.
So, but still means at least 2 weeks time will be necessary to learn how to operate the VCA and let it learn how you want to operate it.
Yes, but I think that the part of the nice thing about the decoder adaptation is that the goal is that we can, even during that kind of 2-week period, we still had an operational interface, right, that he could use to perform the task. And so the goal is to sort of assist, and in some cases actually in brain-machine interfaces, they will use this even like assistive paradigm, where you have kind of a robotic assistant or automated controller that participates along with the subject, and you gradually fade that out over time. And so that is one strategy to kind of make sure that you have an operational interface even while this kind of longer learning process is happening.
So Amy will continue this conversation in the afternoon. I don't want to hijack the dialogue from the audience. Colleagues, please, more questions. Go ahead.
Would it help if we, at the very start, do some sort of mapping and then see if the subject moves the hand from one point to another point, see which neurons fire, and then lay the electrodes along those paths, or somehow sample from those electrodes, those neurons?
Right, so that, in general, no, because controlling, wait, sorry, can you say your question again? I want to make sure I'm understanding.
Ask the subject to move from point A to point B, or think about
move their hand, or
yes.
So all of the examples of smooth batch, for instance, that I showed you, where the initial performance was terrible, the way that we trained that decoder was by having the subject watch a cursor move through trajectories. And so you see patterns of neural activity during that context, but it doesn't relate necessarily to what the neurons do when they're controlling the brain-machine interface. Yeah, there's sort of a very distinct mapping between those two. They're kind of very different contexts. That said, it actually does depend a little bit on, I think, how the subjects are trained. So one really interesting thing is that some people will, in these cases of able-bodied animals, right, some people will have a brain-machine interface task and they allow the subject to keep moving. And what often happens is the monkey keeps moving their arm while they're also controlling the brain-machine interface. And then over time, the monkey realizes, oh, it's not actually my arm that's controlling this. I don't want to move anymore because why exert that energy? And in those cases, often people actually see that neural activity is similar between arm movement and brain-machine interface control. But in our case, what we were doing is asking the subject from the very beginning to not move their arm. And so that potentially leads them to different strategies to control the interface, right? So going back to, what they're doing, maybe, the monkey is thinking about wiggling his foot versus, something else during the task, as opposed to thinking about it from the perspective of moving their arm. And so, the strategy that the subject comes in with and how they approach the problem is going to ultimately influence how this works. And I think going back to the goal of robustness, that's why we kind of need to have a system that allows for all possible strategies, right? Because everyone's going to approach this a little bit differently.
Are your monkeys better than people? Like going back to the BMI literacy problem?
It's an interesting question. I think that it's hard to quantify that really carefully. There's actually a broader issue in the field that there's not a lot of standard metrics. And so there's enough variability between like similar studies that have been done in people and my work that I can't do a head-to-head comparison to quantify it carefully. I would say, generally speaking, the monkeys are probably a little bit better. Some of that might be due to the fact that they have a lot of motivation, whereas the subjects are, they're not paralyzed, but I guess subject motivation varies a lot more from person to person depending on a lot of factors, whereas the monkeys, because we're controlling their motivation by incentivizing them to do the task. And so I think that's an additional factor. Yeah.
Is there any signal on whether or not this literacy could be correlated to the neurons you choose to measure, opposed to the person's ability?
That's a great question. So the BCI illiteracy term actually comes mostly from the non-invasive community. So it's in part, I think, because non-invasive BCIs have been done in a much larger patient population, so they can actually properly kind of estimate the percentages. And that's the context in which it's seen. It's possible that, some of the details of how they're doing the EEG recordings might influence it, but within a given study, there will typically be some portion, where they're controlling, broadly speaking, for how they're making the measurements. There still is a population of people that can't use it. Often that's attributed to the fact that, or people often attribute it to the fact that EEG is just a really broad signal and maybe it's just not a useful signal. But I think it actually, goes back to this issue of how the subjects are learning the interfaces. And I think if we start doing invasive BCI interfaces, we'll start to see people that are, BCI illiterate, quote unquote, even with these more spatially resolved signals. But yes, it probably is related to how we're picking the signals. Yeah.
Is there any kind of thought as far as the degrees of freedom that you have being kind of more natural? Would a more 3D version of this maybe just result in better because it's kind of more natural for people and for monkeys even?
Yeah, I think that's definitely possible. One great study that points to this is that someone kind of went back through a lot of the existing BMI literature and kind of looked at what was working well and what didn't work well in terms of decoders, but from a control perspective. And what they found is that when you have a cursor that kind of is being controlled in a way that obeys reasonable physics, where position is the integral of velocity and, these sorts of things, that performance is better. And so that points to kind of this idea of, having kind of realistic physics and other, properties of the interface are going to be really important for performance. I haven't thought about that in the context of like 2D versus 3D, but I think it makes a lot of sense that sort of the nature of the interface and kind of how similar it is to control schemes that the brain has seen before will probably influence performance. Yeah.
But then birds are actually the only 3D creatures. So human, we're 2D. We live on the upland.
Yeah.
Yeah.
Yeah. Do you think there might be ways to speed up this feedback loop by, rather than going through a complex task, you give like the brain its success?
Right, so like through
Response.
stimulation, you give feedback. Yeah, people have started doing those experiments where you give feedback not just through natural sensory systems, but by stimulating neurons in the brain. It's a really interesting question, and I think we don't yet know whether it would necessarily speed up learning, but we do know that that type of signal can be used to inform learning and to drive control in these systems. So there are examples of brain-machine interfaces where neural activity is being used to control a device, and then the only feedback the subject gets is neural stimulation, and that works. So,
More questions? Shall we thank our speakers? Thank you. Two things.